Um, yeah, I'm happy to talk to you all. I'll go over a little bit of where I work, the Alaska Ocean Observing System, but then move quickly into harmful algal blooms and what they are and all that, and then hit on a few subjects that I hope are particularly interesting for you guys, which is walrus as one of them, and then other impacts on fish and other things. Um, I'm really happy to talk to you because I have realized that really the kind of Bristol Bay region is a region that I have not had a lot of interaction with. And so this may be the first time that I talk to some of you. And so if there's any follow-up questions or anything, please don't hesitate to contact me either through email or phone number, which I'll put at the end of the presentation here. So I'm going to share my screen. Let me know if that doesn't work, but I think that should work. So today I'm talking to you guys about harmful algal blooms in Alaska and how they're a risk to food safety and security. And um, there'll probably be a bunch of questions, which I'm happy to answer either during the talk or at the end, whatever works best for everybody there. Just real quickly about the Alaska Ocean Observing System. So I'm a scientific program manager there. I do lots of different things, but one of the things I do is coordinate the Alaska Harmful Algal Bloom Network, which I'll talk to you about in a minute here. But just generally what we do at AUS is we try to fill the gaps in monitoring and observing on the oceans, and facilitate partnerships to gather more information from the oceans and then serve that data back to communities, to stakeholders, to ocean users, to anybody who might need those data. So we're one of 11 regional programs across the US. And as you can see from the map there, we cover one of the largest marine areas because we have one system for the entire state of Alaska. So some of the things that we do to kind of get those data and present them is we put out assets in the water, so moorings, buoys, gliders, radars, things like that. We partner with a lot of different people in order to get those data as well. So we'll sometimes fund part of someone else's program as long as they provide data back to us to present publicly. We'll also use some money to try to look at new innovative ways of getting some of those data, which may not have been tested in Alaska before. So wave buoys, satellite imagery, things like that. And then we try to show the data in a meaningful way through our data portal, and other data products. And our data portal is very big and unwieldy. And so if anybody tries to look at it and can't really make sense of it, please reach out. We're trying to make it better. But I always want to make sure that people, if they try to go to our data portal and they feel like it's not really accessible to them, to just please reach out to us and we'll help you try to navigate it. And then the other big thing that we do is we try to leverage a lot of our funding into collaborations and partnerships to really get the most out of what the money that we do get. And so we have a lot of collaborative networks that we coordinate. One is the Alaska Water Level Watch, which has water level stations across the state to look at stuff like storm surges and floodings and stuff like that. The Alaska Ocean Acidification Network that talks a lot about the changes in ocean chemistry that's leading to acidification. And then the Alaska Harmful Algal Bloom Network, which I coordinate, and so that'll be the main focus of today's talk. So just talking about harmful algal blooms and really getting to the meat of the talk here, I first always want to make the point that algal blooms in general are not by nature harmful. In fact, in most cases, they're essential, very beneficial to the marine environment, and they're really the base of the food web. And so here's a satellite imagery of the Bering Sea, including the Bristol Bay region in May of 2021. And it's really kind of the peak of the phytoplankton bloom there in the spring that kicks off a lot of the productivity that we see in the Bering Sea. And so all these swirls of cool colors, those are all some kind of phytoplankton that's blooming. And in all these cases, probably they are not harmful. They're very beneficial blooms that are happening. What happens is sometimes some of the species that are blooming may be things that produce a harmful effect. So if you have an algal bloom that leads to a harmful effect, that's a harmful algal bloom. And so here I have one phytoplankton in that picture there, that's an alexandrium that I'll talk about in a minute. If that blooms, that leads to a harmful effect most of the time. The harmful effect can be things like toxins, which is what we're primarily worried about in Alaska, but it can also be things like phytoplankton that have lots of spines and spikes on them that actually can clog up the gills of fish and lead to fish kills without them being actually toxic. They can also discolor the water. They can kill a bunch of animals that then wash up on shore. And the harmful effect is really the unseemly death on the shores. And so Florida deals with a lot of that kind of stuff. But for us in Alaska, it's mostly a toxin issue that we're worried about with harmful algal blooms. One quick thing I want to mention is that people do call harmful algal blooms red tides sometimes. That's what we mean when we say harmful algal blooms is red tides. We tend not to use the term red tides up in Alaska because we don't want to make it seem like if it's not red, there's no problem. Because in reality, most of our harmful algal blooms up here in Alaska do not discolor the water. And so you don't actually get a red tide. When we do get red tides, which does happen, especially in South Central Alaska, like Kachemak Bay, a lot of times you'll have things blooming that don't have a harmful effect, but they do discolor the water a little bit. And so you'll have like mesodinium blooms that have kind of a reddish tint to them. They're actually really great oyster food. So the oyster farmers love to see that. It is technically a red tide, but it's not harmful. So what are the ones that are harmful in Alaska? So here's four of the main ones. Alexandrium, like I just mentioned, that's the primary one we're worried about. It produces a wide variety of toxins, actually. And if those get ingested by humans, can lead to paralytic shellfish poisoning or PSP, which some of you may have heard about. There are a few other ones that produce toxins, dinophysis on the bottom there. Pseudonychia, the top right, both of those are marine as well as Alexandrium. They don't usually lead to deaths, but they do lead to some uncomfortable conditions, such as diuretic shellfish poisoning or amnesic shellfish poisoning, which can lead to temporary memory loss. Mostly in Alaska, we don't see a lot of those. We see a lot of the phytoplankton, the pseudonychia and the dinophysis themselves, but we don't see a lot of the toxins, and we're still trying to figure out why that is. It might come later in the future, but for right now, those don't tend to be toxic in Alaska, even though the phytoplankton do have the capacity to be toxic. And then cyanobacteria, I put the bottom there, the blue-green algae, those are primarily freshwater and estuarine phytoplankton blooms, and they can impact freshwater sources of drinking water, which can be an issue. But we don't see that a whole lot right now in Alaska either. So the main one that we're worried about and I'll talk about today is Alexandrium here. So how can this kind of move through the food web to affect humans? How is this a threat to food safety and food security? Here's just a diagram of a marine food web in Alaska. And basically the harmful algal blooms, they can take the same path as just all phytoplankton can take into the marine food web. So sunlight, nutrients, temperatures all contribute to having a bloom happen. That bloom could be ingested by things like zooplankton, which then can get eaten by fish, small fish, then large fish, then seabirds, marine mammals. Or the harmful algal bloom could be ingested by filter feeders like bivalves, mollusks, and things like that. And that really is the most direct way that it can impact humans because those harmful algal blooms are being ingested by a filter feeder and accumulating in the blue mussel, for example, or the clam, and then those clams being eaten directly by humans is the fastest way that this can lead to a food safety issue. But also, humans potentially eating parts of fish or parts of birds or marine mammals, that can also potentially lead to an exposure to harmful algal bloom toxins. The other thing that we're still learning about is how these toxins impact the health of the animals themselves. So at what point do fish start feeling impacts of having ingested toxins from harmful algal blooms? I'll show you some data for that, but you'll see that there's still a lot of questions, and we don't know. We can't really ask the fish or the birds at what point they start feeling sick. all we can really see is when they show up dead, and then we sample those carcasses and what are the levels of toxins in those tissues. That's the main way that we've gotten information from the impacts to animals. And then, of course, that can lead to a food security issue if those toxins are so intense that they lead to a lot of die-offs, and then the food source of coastal communities could be affected by that. And I always like to show this just as a kind of a PSA, a public service announcement to people about what it is what are some of the symptoms when you start being affected by paralytic shellfish poisoning if you eat foods that have accumulated these toxins. So just to give you like a baseline, this number is going to come up again a few times in the talk, but we start feeling symptoms as humans when we consume foods with parallel shellfish toxin levels that are above 80 micrograms per 100 grams of food tissue. So that 80 number is kind of like what we use as a benchmark for knowing when things are starting to impact us symptomatically in humans. And that can change based on how big the person is, how, obviously how much food they've eaten and all that kind of stuff. But here's the list of symptoms that you can get if you start experiencing PSP. So at first you get some pretty kind of non-specific symptoms like headaches or nausea, which can happen with lots of other stuff. But then there starts to be some more telltale signs of PSP that include things like tingling or numbing sensation, especially around the lips or the tips of your fingers, like extremities. Um, and then when you start getting really into some worrisome symptoms is when you start not being able to move your arms, because that's the beginning of the paralysis part of paralytic shellfish poisoning. And really, where it gets bad is when that paralysis starts affecting the respiratory system. And when people die from PSP, that's usually how they die, is they are not able to breathe. Um, the symptoms, they can come on within minutes if it's a really high exposure level, or within hours, sometimes up to a day. It might take, and usually when it takes that long, the person hasn't ingested a high level, and they're probably going to be fine, just maybe feel some tingling sensation and nothing more. But if that tingling starts really quickly, like within minutes or within an hour or two of eating seafood, that's when it's worrisome because it can progress to being more worrisome symptoms. Cooking, freezing, vacuum packing, drying, none of that seems to remove the toxins. These are relatively stable toxins, unfortunately. And then there's no antitoxin. So it's if you go to the hospital, they won't give you a way to reverse the impacts of the toxins. What they'll do, and why it's really important to seek immediate medical care if you start feeling these symptoms, is they'll provide supportive care, primarily in helping you to breathe. And as long as you can get through the initial stages, um, your body will eliminate those toxins naturally, and you'll be able to survive through it. And as far as we can tell, there are no like chronic long-term impacts. So it's really about acute care, supportive care to get you through the worst of it. And so if they just help you breathing while you're at the hospital, and then you will naturally eliminate the toxins, and you'll make a full recovery. But so that's why the main take home here is seek immediate medical care if after eating seafood you start feeling these symptoms. Unfortunately, sometimes it's so intense that there's no way of having that, people getting that medical care. So the last death of PSP in the U.S. Actually happened in Alaska in 2020. A person in Dutch Harbor, right around July 4th, actually collected some marine snails and mussels and ate them, and they died very quickly due to PSP. If you remember that regulatory limit of 80 micrograms that we use, the blue mussels that were tested that this person ate were 11,000 micrograms per 100 grams. So almost over 100 times higher than the regulatory limit. And so that's why the onset was so quick. And this came soon after there was a relatively warm blob of water around the Aleutians that probably led to an Alexandrian bloom that then got taken in by the blue mussels, and the person ate those blue mussels pretty soon right after that. So it does happen. You know, I always try not to be alarmist in these talks. Like it's not the last one that happened was 2020, so 5 years ago. Since 1993, there's been I think five deaths. So it's definitely not something that happens every year. It's just a high consequence issue when it does happen. Obviously, it's the loss of life is always tragic. So you know, keep. I'd always like to tell people to keep that in perspective. It's a high consequence issue, but it doesn't happen every year. And then there is actual a statewide public response kind of set up for this. So within the state of Alaska Department of Health, the state statutes require healthcare providers and labs to report specific diseases and conditions. And here's the list there, and you can see paralytic shellfish poisoning is one of them. And so that's why if you start feeling symptoms and you go to the clinic, it's something that they need to report to the state, and the state will do an investigation of the situation to make sure it is paralytic shellfish poisoning, and to do some additional testing. But that means that there is a state Department of Health kind of response plan in place for this condition. I'll make just a quick side note here on commercial shellfish. This comes up as a question sometimes. So for commercial shellfish, which is primarily oyster farms in Alaska, primarily in South Central and Southeast Alaska, the Alaska Department of Environmental Conservation does closely regulate the commercial sector, where every time a farmer wants to harvest their crop and sell them, they have to send a sample to the lab in Anchorage that will then test the sample, make sure it's not above that regulatory limit. And if it is, then they have to go through a whole process of keeping the oysters or whatever the product in the water, doing testing on a regular basis until the oysters naturally get rid of the toxins, and then they can reopen and sell their product again. So there's been no reported human cases of PSP from commercial product in Alaska. But one thing to note is that that is just for the few farms of commercial farms in Alaska. There's no equivalent state monitoring or regulation of subsistence or recreational harvested foods. Okay, so how HABs happen? Primarily, I mean, this is basically a plant that lives in the ocean. So temperature, nutrients are the two big ones, and sunlight are really the main components that lead to a bloom happening, a bloom of of any phytoplankton. The nutrients one is interesting because a lot of harmful algal bloom species like high nutrient levels. And so for us, the limitation is primarily on temperature, and to some degree sunlight when you get into Arctic areas where there is a lot of sea ice cover, which can actually keep the hot HABs from happening. So temperature is the one that we really look at a lot. If it's above 8 degrees Celsius is when we start being worried about the fact that we have conditions that are favorable to blooms. And for Alexandrium specifically, they have an interesting a life cycle where they have free floating cells. Those are the ones that produce the toxins primarily. But they also have a cyst stage, which is the part here that's inside in the sediment. And these cysts, they can lay dormant in the sediment for a long time. And then when conditions are right, they can then germinate and go into the water column and then produce the blooms that are toxic. And so knowing where those cysts are is a big part of the story here. And so here's a map here from a very big professor that works on HABs, Don Anderson. He's from Woods Hole Oceanographic Institution on the East Coast, but he's worked a long time in Alaska. And his group has mapped out the sediment cysts across a lot of Alaska, mostly northern Bering Sea and north of that. But they're starting to do more work further south in the Bering Sea because they're realizing that there's more and more regions with these cysts. But the problem with these cysts is that they're there, as far as we know, they can kind of live indefinitely in the sediment. And we probably won't get conditions in the Chukchi Sea being conducive anytime soon. But at some point in the future, water temperatures might be warm enough for that huge cyst beds, that big red area, in the Chukchi Sea here, to start blooming. And in that case, it would lead to a lot of potential issues. And the problem with temperature here is that the warmer something is, the fewer days is required for these cysts to germinate. So this relationship here that you can see is from a bunch of data points in the field, but basically if your water temperature is 2 degrees Celsius, which it is in a lot of places in Alaska, that means that you're looking at something like over 60 days until germination. So you need to have 2 months of 2 degrees Celsius water for these cysts to germinate. As opposed to if you get to water temperatures that are 8 degrees, now you're talking more something like 10, 12 days. And so the likelihood that the water temperature stays 8 degrees C is pretty low right now, but in the future, if that gets warmer, then we may have germinations that are much more likely in the future. So here's a map from ACAP from their publication on Alaska's changing environment, and you can see like how things have gotten warmer in the past. I think this is 1982 to 2024. And so you can see some changes here in the water temperatures. Some areas like Kotzebue Sound and the Yukon River Delta being much warmer, but even Bristol Bay here having a couple of degrees warmer Fahrenheit than 20 years ago. That if those trends continue, that could just mean more opportunities for blooms to happen in the future. Yeah, so higher temperatures are more conducive to germination blooms, and then also higher temperatures lead to longer ice-free periods, which means longer period where the sunlight can penetrate into the water. So these blooms happen. The toxins are produced by the blooms. How are we learning about how these toxins are moving through the food web? Well, a lot of that is just sampling more and more items from the ecosystem to see how they're accumulating toxins. So here's figures from a paper from another researcher that does a lot of work up in Alaska, Kathy LaFay. And here's some of the animals that she looked at. So clams, worms, bowhead whales, walruses. And you can see they're color coded here by different levels of toxins that were tested in their tissues. And so some of the clams in the areas where there's those big cyst beds are pretty high in their toxin levels. And I know that walrus are a particular concern in Bristol Bay, as well as salmon, obviously. I'll talk about salmon in a minute, but there's a new paper that just came out, so I thought I'd highlight it on the impacts on walrus. It's still very much being researched, but this paper that came out this year by Kathy LaFay's group, actually, same group of the figures I just showed, they looked at a modeling approach where they looked at the toxins that were present in Alexandrium, the toxin levels of Alexandrium, and how those prey were ingesting those toxins. So knowing the toxin level of the preys, and then figuring out what the consumption rate of walrus were on those specific preys, and therefore how much toxin could be transferred up to the walrus, and at what point the walruses start feeling impacts from those toxins. So this second figure here in this paper kind of shows the different areas where they looked at the clams primarily, but also snails and worms in these different regions, and then they modeled kind of like general areas where those animals were of different had different levels of toxins. So the red and the orange are higher levels of toxins in the prey species, and therefore the danger, the health risk to the walrus are higher in those areas. And you can see especially, so, and this is for two specific blooms in 2022 and in 2019. You can see in 2019, this area around Ledyard Bay, where there's a lot of red, a lot of prey items that were high on the toxin levels. That's also an area where a lot of these tagged walruses from Point Lay were going out. Those little black dots are tagging or locations of the walrus that were tagged, going out into that specific area and harvesting, eating clams out of those potentially high toxin areas. And so basically, the conclusion that this paper comes out to is that walrus are potentially impaired during these blooms to levels that we know can potentially impact walrus. It may not always kill them, but it could impact their feeding behavior, their mating behavior. And so models like this could be used in the future to assess future risks. If we know the toxin levels of different food items, we might be able to find out what the risk is to animals like walrus. And there's lots of other animals that are being looked at, but walrus is one of the first ones that this group studied because of the importance of walrus to a lot of communities. Okay, so how do we kind of try, as a state, as a group of organizations trying to help with this issue? How do we try to minimize the risks to food safety and food security? Um, uh, in our minds, there's like kind of three approaches that are needed simultaneously. One, we need to monitor the algae, to see whether or not there are harmful algal blooms in the water. So what organisms are in there? Are they harmful species? It's great for understanding bloom dynamics, figuring out what environmental parameters lead to blooms in different areas. In and of itself, though, it cannot determine the safety of food items. So just because you see a bloom doesn't mean that food items are going to be toxic. And just because you don't see a bloom doesn't mean you didn't miss it, and doesn't mean that foods can't be toxic. So really, to learn more about food safety or the amount of toxin that are in the foods, you really need to do the testing of the foods themselves, of the food items themselves. So you can sample, a wide range of different organisms that can be tested, both food items that people eat as well as other species in the ecosystem. It does require labs and sometimes some pretty advanced techniques. It's not the kind of thing you can do on the beach. That's not a dipstick test. It's something that requires relatively advanced techniques and sometimes pretty expensive equipment. Um, but if you can get the results back to people quickly, you could potentially use that as a way of aiding in deciding whether or not to eat certain foods. And that's what we're trying to set up, and what has been set up by some groups in different parts of the state. Primarily Southeast Alaska has done a good job having a lab locally that can test quickly so that people can decide whether or not they eat the harvest that they make. And then the third leg of the stool is communication and outreach. And that's really important. Doing these kinds of talks that I'm doing now to just let people know about the issue and what can be done. But also during a bloom or during a toxic event, making sure that information gets out to the regional communities so that they can maybe limit their behavior, their harvesting or their consumption, and limit the impacts that those toxins might have on other people. So just having contacts, people like the Sea Grant map agents in the different communities, that they can receive that information and then know how to distribute it locally, is a really important piece of this puzzle. So an example of how kind of these three steps kind of came together. Um, in 2022, there was an Alexandrian bloom that was caught by a research cruise. And here's the map of the stations, and the dots there are the stations that were sampled for phytoplankton. And the more red the dots are, the higher the Alexandrium density was. And so you can see around St. Lawrence Island here some really high dense stations where there was a lot of Alexandrium. And so before the cruise, there was discussion about how kind of people were going to communicate from the cruise to the communities. And so there was a bit of a regional response communication network that was set up through a lot of work and collaboration. And that allowed this information to get out to people. And so through both the Nome Nugget and the KNOM radio station, that information that there was an Alexandrian bloom in the area was communicated to the community, or to the various communities. And I think about a month later, somebody actually got a surf clam, an Arctic surf clam, on their halibut longline that came up. And because they had heard of the story, they actually sent that surf clam in for testing rather than consuming it locally. And it came out after testing that this clam had a toxin of 450 micrograms per 100 grams. So remember that 80 is kind of the regulatory limit where we might start seeing symptoms above that. So 450 is pretty high. It's not astronomical. There's a chance that if like an adult were to eat this clam, they would maybe feel a little bit of symptoms, but nothing bad would happen. But the really scary thing is that this family said that if they hadn't sent this in, they probably would have fed it, because it's a delicacy, to their elders and to their granddaughter that was a baby at the time, and that could have actually led to some really dire consequences. So, through the work that was being done and through this communication network, a potential, really bad situation was avoided. And so that's kind of where the Alaska Harmful Algal Bloom Network comes in. It's just a way to put in place this communication, this support for testing, for additional information to come in, especially in a state like Alaska that's so huge. You just need people to help each other out, to get the word out, and to do more work in more places. So the network was kind of formed as, it's an informal network really, but it was formed in 2017. It's just a platform really to communicate and support have monitoring, research, and outreach across Alaska. I put some of these are just a handful of the organizations that are part of the network throughout the state. Um, and basically it's really these organizations that do the work on the ground. And so the strength of the network comes from the members that are doing the work, and then all contribute to kind of a shared goal of communicating and sharing that information across all the different organizations. Here's just a list of a more exhaustive list, although I think even this list is probably a little bit out of date. I need to update it, but yeah, there's over 60 organizations now that are part of the network. And see are the kinds of work that these AHAB network members do. So the Kachemak Bay National Estuarine Research Reserve, for example, they do regular phytoplankton monitoring across a wide variety of communities that all send them samples. And then they go through and through the microscope, they look at to see if they see these different HAB species. And this is just an example of what they produce. So the green bars are the weeks in which they saw these different HAB species show up in their samples, not always at a bloom level, but just present as even a single cell. The Southeast Alaska Tribal Ocean Research Consortium has been doing a lot of great work for over 10 years now. And through the lab that they established at the Sitka Tribe of Alaska, they've gotten a lot of toxin data back from a variety of food items, primarily clams and mussels. And they share that immediately to all of their communities around Southeast Alaska to tell them whether or not certain areas have toxins in their foods or not. And so you can see here a timeline of all the toxin test results that they've gotten. Here's the salmon and other fish results. So some federal agencies like NOAA do a lot of this work. So this is primarily from around Kodiak and Kachemak Bay, but it seems to be that salmon and Pacific halibut don't accumulate toxins at a very high level, at least from the samples that I've gotten so far. And when they do accumulate it, you can see that these graphs are divided up by the tissue types. And so digestive organs, liver, kidney, muscle, roe, and stomach contents. And anytime you have a sample above that red line, which is the regulatory limit again, it tends to be in organs, either in liver or kidney, and sometimes I think in digestive organs, but really is it in muscles. So it doesn't seem like, for salmon at least, that these HAB toxins accumulate very quickly in muscles. Or it might be that the accumulation is faster in these organs, and before the toxin level gets too high in the muscle, it kills the fish because of the toxins in the organs. So that might be the dynamic that's at play here. But it seems like the accumulation is not as quick as in the muscles as it is in other tissues, for salmon at least. And then other federal partners like USGS, they do a lot of work on marine mammals as well, but also on birds. And you can see here just a bunch of birds being tested here for the presence of saxitoxin. And so the red bird symbols are the ones that have gotten tested and shown up to be have toxin levels in their tissues. And so you can see it's not every bird, but birds throughout the places where they've tested them have been shown to have toxins in them. One of the limitations that we have in Alaska is that we have a very big state, first of all, but we don't there's not a lot of places that have these labs that can test for the toxins. So we have the main lab in Anchorage, that is the state lab. That's the only FDA approved lab for food safety. So that's where all the commercial samples go. In the previous few years, we've had a couple of other labs, mainly at tribal organizations like CTOR I mentioned, but also Ludic Pride Marine Institute in Seward, that have active toxin testing labs. They're not technically FDA approved, but they do. So they can't they don't have any authority to say whether something is safe to eat or not, but they do produce good data for people to use, or not use, depending on what people want to do. They can't tell people what to do, but they can provide them with the information. And there's also other labs that are outside of Alaska that test a lot of Alaskan samples. So USGS, NOAA, and the Woods Hole Oceanographic Institution. Sea Grant has a lab that's being developed in Kodiak, which is really great. And there's one that's being considered in Nome, which would be really helpful for Western Alaska. But there's other places that could have potential labs. In Dutch Harbor, on the Pribilofs, they're actually on St. Paul, they're starting to develop a lab there. And then Kotzebue is going to get a lab pretty soon, and maybe up in Nukuavik at one point would be great to have one too as well. So we're trying to develop these capacities. But right now it is a little bit limiting. And if somebody wants something tested, they have to send it at least to Anchorage, if not further, to have it tested. And that is a vulnerability that we have. And where the commonly tested species that people test in these labs are a lot of bivalves, but they can do fish, they can do other invertebrates as well. Okay, so I'm gonna try to finish up here fairly soon, but just what is the emergency response kind of needed when something shows up at one of these labs as being toxic? Like I said, communication is key. And so if like a bloom of Alexandrium is detected, we communicate to nearby communities to let them know, hey, there is a bloom. And then we try to communicate with DOH so that they can let their clinics and hospitals know, here's, a PSP, be on the lookout for PSP symptoms because there's a bloom happening, for example. And definitely if somebody detects high toxin levels, then we can get DOH directly involved and again, communicate with people in the area and at least suggest suspending harvesting in the area to people. The last thing I want to mention is just on this little piece that AUSE does do. So like I said, most of the network is doing lots of work kind of regionally, as well as coordinating the network. I run this program for imaging flow cytobots, which are ways of detecting the presence of HAB species. So this is what it looks like here, and it basically just takes water samples, and it's like a microscope, an automated microscope inside it. Takes water samples and then puts the water through a flow cell that takes individual pictures of individual particles. And then you can look at those images and see if there's any species of harmful algal blooms that you should be worried about. So here's kind of how it looks on the inside. And then, I need the sound. But as it's running, you can see it takes pictures of individual particles as the particles are running through the flow cell. So each of those red squares is an image being taken of something running through the IFCB. So here's what kind of the output looks like. So this was on the U.S. Coast Guard cutter Healy in 2024. I had this IFCB from Dutch Harbor all the way up to Canadian waters and then back to Nome. And so here's the kind of output that you get. So pretty pictures of phytoplankton. So this was a sample that was taken in the Chukchi. Here's a different sample. You can see it looks very different. The phytoplankton community near the Mackenzie River Delta. Here's a sample in the Bering Strait region. And then here was a sample early on in the cruise, right in the middle of Southeast Bering Sea. And the thing that we were looking for and hoping not to see was this. And all these dark circles there, those are all Alexandrium cells. And so this is a pretty high density. I can't remember what this one was. I think it was something like 4,000 cells per liter, which is getting pretty high. And so when we saw this, we then drafted an advisory and sent it to the communities that I had contacts for at the time in the region. And so now both Dillingham and Bethel are on my list of people to send advisories to. But in 2024, they weren't yet, unfortunately. And so here's a zoom in of the data. So again, each dot here is a sample, and the color of the dot is the density of Alexandrium. And you can see this area here was so it was on July 5th, about 160 nautical miles east of St. Paul that we saw the peak of Alexandrium densities. 4,600 cells per liter was the max. We start getting worried at 1,000 cells per liter and really worried at 10,000 cells per liter. So here we were at a worried state where we still wanted to let everybody know, but we didn't expect this to accumulate really, really high levels of toxins. And 6 of the samples in this transect were above 1,000 cells per liter. All the little arrows here are the currents, and you can see that just the patterns of the currents are kind of all over the place. So at the time, it was kind of hard to determine whether this bloom was going to go towards Bristol Bay or towards St. Paul. We just didn't know what was going to happen because it was so complex. But so like we said, we'd send out an advisory to as many people as we could at the time. And lo and behold, about a month later, there was a mortality event on St. Paul where fur seals and fish washed up dead on Benson Beach here in St. Paul. And then we looked at a particle tracking model to see where those particles went after they were detected here in the Bering Sea. And you can see that as you work through this chronology in the colors here, the particles went east or west and then north right around St. Paul. And so this is a paper that we wrote after this event happened to kind of explain the process of all these different steps happening. Oops. And then in 2025, I had another IFCB in the region twice during the summer of 2025. And again, around the same area, we saw some more high levels of toxins, of Alexandrium cells, not necessarily toxins, in the Bering Sea again. So this is just where we had samples taken, and it's obviously not exhaustive of the area, but we did see it again in July and early August around the Southeast Bering Sea. And this time, about a month later in mid-August, we got a marine mammal and seabird and fish die-off on St. George this time. So we need to look at that closer. We responded as best we could at the time, but we need to do a retroactive analysis of what happened there, just like we did for the 2024 bloom. And just to show you, like, this is some of the research vessels in any given summer in Alaska. And so there's potential for a lot of these to be carrying these IFCBs and potentially being kind of a background data collection method for HABs across most of Alaska. But we could also put these IFCBs in specific locations. And you can see in both of these situations, Bristol Bay isn't very well represented here. So anybody that has any suggestion as to how we can get more samples, more data out of the Bristol Bay region, I'm always all ears if anybody has any interest in being part of this work. So last slide here, just things that we need to do, increasing just efforts on monitoring for both the phytoplankton and for sampling for toxins. And that includes getting more labs up and running, which will be a multi-year effort, but we're working on it. Try to deploy new technologies. So these imaging flow cytobots, like I said, but also we're working on trying to get satellite imagery to be more informative than it's currently, to try to detect large blooms kind of more remotely. And then the big thing is making sure that coastal communities that want to work on this and collect this kind of data, that they're funded, that they have people that can get the samples, that can do the phytoplankton monitoring locally if that's of interest to them. And so that's definitely including the Bristol Bay area. If anybody there wants to be involved in this work, let me know and we can try to find ways of supporting you to do that, whether it's getting microscopes or paying for testing for samples and that kind of thing. And that's it. So there's my contact info. We also have a website, Alaska Ornithological Bloom Network website. Feel free to email me or give me a call. And hopefully I have time for questions. I'm happy to stay on a little bit past one, but definitely contact me if you have any questions that aren't answered today. Thank you very much.
Thank you very much. I'm going to ask if online or in the room we have any questions. Yes, I got one here.
Yeah.
This is Janelle Trowbridge.
To talk louder.
This is Janelle Trowbridge. I'm actually a wildlife biologist in the region for Fish and
Oh,
Wildlife Service. So
yeah.
we've probably met online at some point, but I'll probably follow up with you.
Yeah.
I do watch seabirds and walrus, so I do have curiosities. But my kind of two questions are, how long does it take for the shellfish to move the toxins through them after a bloom and after that bloom has moved away? And then could you also get poisoning if you're below that 80 number, but eating like, 20 shellfish in a day or something like that?
Yeah, so excellent questions, both of them. So your first one is well, another reason why this is a complicated just situation to monitor, because different species will both accumulate and get rid of toxins at different rates. And getting rid of toxins is called depurate, the depurate or the depuration rate, just in case you ever see that. And so even within like invertebrates and bivalves, blue mussels are actually known to both accumulate and depurate very quickly. So in a matter of weeks. So they're very good sentinel species to see whether or not when the bloom was exactly, because they'll give you a nice little peek if you look at a time series. But more worryingly, things like hardneck clams, surf clams, butter clams, they probably keep the toxins much longer, probably on the order of months, maybe up to a year. And so that makes it very difficult, because usually if you don't have a bloom, you think you're probably safe. But if you had a bloom months ago, even into December, sometimes butter clams could still have some toxins in them. Um, and so that kind of leads a little bit to your second question. So the limit or the toxin testing that we do provides you with a measure of the amount of toxins per amount of tissue. So, but really what's the important thing for human health is how much total toxin you've ingested. And so you're right that a low level toxin, if you eat a lot of them, you'll get more total amount of toxins ingested into your body. Um, and so that's why, and it's the same thing for things like mercury. That's why there's regulations as to how many times a week we recommend, pregnant mothers to eat tuna, because we know tuna is pretty high in mercury. So it's trying to limit the total amount of mercury that's being ingested. Same thing here, but the 80 microgram per 100 gram level is pretty conservative. And if you eat one clam that's at that level, or even at like 100, you're probably not even going to get any symptoms. But we have that level of conservative in there as a level of safety, so that if somebody eats a pound of mussels and they're at that 80 limit, that's you might get some symptoms, but you're probably not going to die from that. Um, so yeah, so definitely the amount that you eat is impactful, but the regulatory limit kind of tries to take that into account a little bit. And the other thing I'll say, just because you're a Fish and Wildlife Service, the other thing that complicates all of this is that HPAI, the high path avian influenza, does also kill birds as well, and HABs also kills birds. And so a dead bird, you don't know if it's that or if it's something else. And the problem with HPAI is we don't want people to put themselves in danger by collecting dead carcasses of birds necessarily. And so that can complicate the ability of getting samples to test them for harmful algal blooms. So people like Fish and Wildlife staff that are kind of trained in proper protocols for collecting samples is really great to have locally, because they can get samples even if HPI is a potential cause of death.
Great, thank you.
Yeah.
Any questions online or in the room? Well, I think that wraps that up. Thank you so much for presenting at the Bristol Bay campus's Lunch and Learn.
Yeah, no problem. Thank you so much for having me. And yeah, I'm happy to also send this presentation as a PDF if anybody would be interested in having that.
Great. And we'll stop the recording and it'll be posted online for others to view.
Perfect. Great. Thank you so much. And don't hesitate to contact me if anybody has any questions.